Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall talk about the synthesis of the non-essential amino acids. Just a quick reminder, we are still in these last few videos, we have talked about the postprandial state. So today, the synthesis of amino acids, we are still talking about this postprandial state that happens after a meal. So in the postprandial state, there is glucose, a lot of glucose that is available from the diet, and that glucose can provide ATP, energy. So there's lots of energy for all the cells because your blood glucose levels are high, and since your blood glucose levels are high, the insulin levels are high. And insulin is an anabolic hormone. It stimulates synthesis. So there's a lot of synthesis going on. Okay, so there is glycogen synthesis that we talked about earlier, fatty acid synthesis, cholesterol synthesis, triglyceride synthesis, and we talked about this in previous videos. So today we shall look at the synthesis of the non-essential amino acids. Of course, the essential amino acids come from the diet. So together with the essential amino acids and the non-essential amino acids, we should have all the amino acids necessary to make all the various proteins found in all of our cells. We shall see in a few seconds that the main street, that's the conversion of glucose, eventually giving you ATP. Main street that not only supplies energy, it will also give us the intermediates necessary to make the non-essential amino acids. And these intermediates you'll see come mostly from glycolysis as well as the TCA cycle. So recall the basic structure of an amino acid. So unlike carbohydrates and lipids are very rich in carbons and hydrogens. Amino acids have, all the 20 amino acids have the amine group, the amino group. So there is a nitrogen that is present. So besides carbon, hydrogen, and oxygen, we now have to deal with nitrogen. So a good way to understand the metabolism of amino acids is to divide the amino acids into two parts. The carbon skeletons, shown here, and the second part would be the amino group. So the carbon skeletons necessary for amino acid synthesis comes from main street. So for example, here is 3-phosphoglycerate, which is an intermediate in glycolysis. This provides the carbon skeletons for making serine and glycine. Pyruvate gives us the carbon skeleton to make alanine. Alpha-ketoglutarate in the TCA cycle gives us the carbon skeletons necessary to make glutamate, and from glutamate we make glutamine as well as proline. And then oxaloacetate in the TCA cycle, part of your main street, gives you the carbon skeletons to make aspartate, and from aspartate we make asparagine. Okay, so the carbon skeleton part of all these non-essential amino acids such as proline, glutamate, glutamine, aspartate, asparagine, alanine, glycine, and serine, they all come from main street. So the next question, obvious question would be, where would the amino group come from? Where would this part come from? So one possibility is this enzyme called glutamate dehydrogenase that is found in the liver as well as brain, kidneys, testes, and other tissues. Now this enzyme catalyzes this reversible reaction shown here where glutamate can be converted into alpha-ketoglutarate and ammonium, or in this direction we have alpha-ketoglutarate and ammonium giving you glutamate. So since this reaction has a delta Z 0 prime of that is positive, high positive plus 30 kilojoules per mole, so it suggests that this reaction should therefore occur in this direction, which means the ammonium could be added to the carbon skeleton to give us the amino acid. So that suggests that we could make amino acids from this ammonium using this reaction. However, in the disease called hyperinsulinism / hypoammonia, this is HIHA. So in this disease, these patients have high levels of glutamate dehydrogenase. Now, at the same time, they also have high blood ammonium levels. So that means if this enzyme is high, if your GDH is high and ammonium is high, this implies that the enzyme is working in this direction. It is used to break down glutamate into ammonium and alpha-ketoglutarate, and not this direction where ammonium can be used as the amino group for amino acids. So another possibility is that we require preformed amino groups. We need this from our diet so that we can make our non-essential amino acids. And this probably comes from glutamate because we do have a taste receptor for glutamate. This taste receptor is called umami. So the fact that we have umami taste receptor for glutamate might imply that we need glutamate in our diet to give us the amino group for synthesizing the non-essential amino acids. There are certain foods that are very rich in glutamate. For example, seaweeds, tomatoes, dried tomatoes, cheese, soya sauce, green tea, and certain mushrooms. These are very rich. They have more than 1,000 milligram per 100 gram of food. So to summarize, the carbon skeleton comes from main street, while the amino group mostly will come from this amino acid glutamate that we get from our diet, and a little bit might come from this glutamate dehydrogenase reaction. So let's look at the synthesis of the non-essential amino acids, and we'll also talk about two important coenzymes, pyridoxal phosphate, PLP, and tetrahydrofolate, THF. So synthesis of the non-essential amino acid alanine. So we get the carbon skeleton from pyruvate, and we get the amino group. This amine group here comes from the amino acid glutamate. This reaction, the synthesis of alanine, is catalyzed by the enzyme alanine transaminase. So this is a transaminase enzyme which is part of the transferase family, and this reaction here is called a transamination reaction. So this is a transamination reaction. So what's a transamination reaction? In a transamination reaction, an amino acid will provide the amine group to a keto acid, and in this case the keto acid is pyruvate, the amino acid is glutamate. The amino acid will become the alpha keto acid, so glutamate becomes alpha ketoglutarate, while the pyruvate, the alpha keto acid, will become the amino acid, in this case alanine. Transaminases require a coenzyme pyridoxal phosphate, or PLP, in order to transfer the amino group from the amino acid to the alpha keto acid. This enzyme, ALT, alanine transaminase, is very rich in the liver, so it's actually used in the hospitals as a liver test. So serum ALT actually helps us monitor any damage to the liver cells. Aspartate synthesis. So once again, now we have the carbon skeleton, and this carbon skeleton is coming from an alpha keto acid, which is oxaloacetate. So the carbon skeleton is from the alpha keto acid oxaloacetate. The amine group, once again, is from glutamate. So the glutamate amine group provides the amino group for aspartate. The enzyme that catalyzes this transamination reaction has several names: glutamate oxaloacetate transaminase, or GOT, also called aspartate aminotransferase, also called aspartate transferase. Different names for the same enzyme. And once again, being a transamination reaction, this transaminase enzyme requires vitamin B6, which is pyridoxal phosphate, as a coenzyme. Asparagine is made from aspartate. Since aspartate gets its carbon skeleton from oxaloacetate, that's the same for asparagine. The amino group comes from glutamate, so this amino group comes from glutamate. The second amino group that parts form the amide linkage in asparagine, that comes from the amino acid glutamine. So we make asparagine from aspartic acid, and this enzyme here is called asparagine. So this enzyme here is asparagine synthetase, which is part of the ligase family. So you can see this amino acid glutamine plays a very important role in the synthesis of other amino acids such as asparagine. Synthesis of glutamate. Now glutamate gets its carbon skeleton from alpha ketoglutarate, while the amine group can come from many different amino acids, because this enzyme that catalyzes this reaction could be one of many different transaminases. So once again, the amine group comes from an amino acid. It's transferred to the alpha keto acid. The alpha keto acid will become the new amino acid, while the amino acid will become an alpha keto acid. So this is a transamination reaction catalyzed by transaminases, and they require pyridoxal phosphate. The amino acid glutamine is synthesized from glutamate, since the carbon skeleton of glutamate comes from alpha ketoglutarate. So the carbon skeleton comes from alpha ketoglutarate. So glutamine also will have the carbon skeleton from alpha ketoglutarate. This amine group of glutamate, so this amine part of glutamate, as we discussed earlier, comes from various amino acids. So that would be this amine of glutamine can come from various amino acids. However, the amine that is part of the amide linkage in the side chain of glutamine, this comes from ammonium. So while the carbon skeleton of glutamine comes from alpha ketoglutarate, the amine groups of glutamine comes from various amino acids as well as ammonium ions. Most of the ammonia in our body comes from metabolism by intestinal bacteria. Some of it also comes from the breakdown of amino acids. Now, glutamine is the most abundant amino acid in our bloodstream. It forms about 20% of the total amino acids that we see in our bloodstream, and it's responsible for carrying the ammonium from the muscles and the brain to the liver and kidneys for subsequent excretion. Now, ammonia in the brain, excess ammonium in the brain is very, very toxic, as we shall see in many different examples in future videos. So it's very important that there should not be any excess ammonium in the brain. So this enzyme glutamine synthetase that is responsible for synthesizing glutamine from ammonium and glutamate helps get rid of any excess ammonium from the brain to the liver, where it's converted to urea, or to the kidneys, where the ammonium is excreted directly into the urine. Glutamine is very important for the synthesis of many amino acids, as well as for the synthesis of the purines and pyrimidines in nucleotides, and other biomolecules such as glucosamine. So most of the glutamine is synthesized by our body because the dietary glutamine is basically metabolized in the intestine. So the enzyme that catalyzes the reaction between glutamate and ammonium to give us glutamine, this enzyme is called glutamine synthetase, or GS, and synthetase is part of the ligase family. Now, there have been a few cases reported in literature where patients did not have this enzyme, glutamine synthetase. So this enzyme wasn't present, or was present in very, very low amounts, and that, of course, led to the decrease in blood glutamine levels. The blood glutamine levels was very low, and interestingly, there was a lot of ammonia in the blood, hyperammonemia. So a lot of ammonia in the blood, and high ammonia levels, as I mentioned earlier, are very toxic to the brain. So so the brain underwent a lot of encephalopathy or pathological changes in the brain. There were 3 cases reported. 2 of them died within a few days. 1 patient lived for around 6 years. So that shows how important it is for humans to get rid of any excess ammonium, which can lead to brain damage and eventually multiple organ failure. Now, it should be noted that glutamate is not only an amino acid used for protein synthesis, it's a neurotransmitter in the brain. So this reaction here can help get rid of any excess glutamate neurotransmitter in the brain by converting it into glutamine. Synthesis of proline. Proline is also made from glutamate, and since it's made from glutamate, the carbon skeleton for proline is also the same as glutamate, and that's alpha-ketoglutarate, while the amine groups of proline is the same as glutamate, which would be derived from various amino acids. Now, in the conversion of glutamate to proline, notice the first reaction. There is a kinase enzyme that adds a phosphate from ATP to this carboxyl group of glutamate to give you glutamate 5-phosphate. And then there is a redox reaction catalyzed by a dehydrogenase. And notice that this group here, this becomes reduced to an aldehyde. So now this structure here has an amine and an aldehyde. So we mentioned several times in previous videos that One reaction that can happen very well in water is the reaction between an aldehyde and an amine. And that's exactly what happens in the next step, where the aldehyde reacts with the amine, not even needing an enzyme because this reaction happens very fast. So this is a non-enzymatic step that gives us this product here, which is then reduced once again by a reductase to give you the non-essential amino acid proline. Now, synthesis of serine, so the carbon skeleton of serine, this carbon skeleton comes from the main straight intermediate, 3-phosphoglycerate, which is part of glycolysis, while the amine group, this amine group comes from glutamate. So in this conversion of 3-phosphoglycerate to serine, we can see the first reaction is a redox reaction where the hydroxyl group is oxidized to a ketone to give you 3-phosphate hydroxypyruvate, and then this alpha keto acid undergoes a transamination reaction with glutamic acid. The glutamic acid becomes alpha ketoglutarate, while this 3-phosphohydroxypyruvate becomes 3-phosphoserine, becomes this amino acid 3-phosphoserine. And in the next step, this phosphate of 3-phosphoserine is removed by a phosphatase enzyme to give you the amino acid serine. The amino acid glycine is made from serine, and since serine uses 3-phosphoglycerate or gets its carbon skeleton from 3-phosphoglycerate, therefore glycine also gets its carbon skeleton from 3-phosphoglycerate. The amino group of serine comes from glutamate, so the same thing in case of glycine, since glycine is made from serine. So the enzyme that catalyzes the conversion of serine into glycine, this reaction here, this enzyme is called serine hydroxymethyltransferase, and this enzyme also requires the coenzyme pyridoxal phosphate. This enzyme also requires another coenzyme called tetrahydrofolic acid or tetrahydrofolate. So the THF basically will help remove this one carbon unit, this methylene unit CH2. So the CH2 unit will be taken up by THF, and the THF becomes N5N10 methylene THF, water is removed, and the serine is converted into glycine. THF is a very important coenzyme in the metabolism of not only amino acids but nucleotides that we'll talk about in the future. THF participates in the transfer of one carbon units that are in various oxidation states. So the one carbon unit could be in a very highly oxidized form, such as formyl. It could be in the methynyl state. It could be in the methylene state, or it could be in the most reduced state, which would be the methyl state. So any one carbon units that are in these states here could be removed by THF from the substrate. So that is how we make these non-essential amino acids: proline, glutamate, glutamine, asparagine, alanine, glycine, and serine. Now notice that we did not talk about cysteine and tyrosine, and that is because cysteine really comes from an essential amino acid, methionine. So since it comes from an essential amino acid, cysteine should really be considered an essential amino acid. Similarly, tyrosine is made from an essential amino acid phenylalanine. So tyrosine should therefore also be considered an essential amino acid. So we have the synthesis of the 8 non-essential amino acids. That is all for now. Until the next video, thank you for watching. Have a happy and a very healthy day.